Hello, Mr. D. This is going to be my presentation on the paper I reviewed, Phylogenomic Insights into the Cambrian Explosion. The paper, Phylogenomic Insights into the Cambrian Explosion, the Colonization of Land, and the Evolution of Flight in Arthropoda by Wheat and Wahlberg, aim to determine the most probable scenario of the evolution of Arthropoda and to give a definitive estimation on their colonization of land and their evolution into flight, utilizing a phylogenetic approach. The two major scenarios they are comparing in the study are the short fuse scenario, where short dramatic divergent events happen during the Cambrian period, and the long fuse scenario, where the time of divergence extends further back into the pre-Cambrian period. Their phylogenomic dataset consists of 112 taxa and 62 genes. This data was partitioned into 6 subsets of 21 taxa for every 27,984 base pairs, as it was a minimum taxa required to implement their A calibration points. A standard setup was then used on the data to compute, and a Bayesian analysis was the sole technique employed to elucidate the phylogenetic relationships. It was observed that 95% of Arthropoda's crown group was all within the cryogenian period, estimating arthropods to be approximately 706 million years old. This data then led researchers Wheat and Wahlberg to reject their alternative hypothesis in favor of the long fuse scenario, as the data suggested a pre-Cambrian origin of the beginnings of Arthropoda. By using their data, they hypothesized that there were 3 separate colonization events on land, all beginning first with the ancestor to myriapods during the early Cambrian, approximately 538 million years ago. Arachnida followed after, around 501 million years ago, and the last clade to colonize land was Hexapoda, approximately 433 million years ago. Although flight was thought to evolve during the Carboniferous period, the data gathered by Wheat and Wahlberg found that the first instance of the evolution of flight came out beforehand, when the ancestor to Pterygota derived from Zygentoma at the end of the Devonian period, approximately 384 million years ago. When comparing Wheat and Wahlberg to other studies around the origin of Arthropoda, such as Regier and co-authors, analyses such as the maximum likelihood analysis was omitted, as Wheat and Wahlberg solely relied on the Bayesian analysis to interpret their data. However, Yang and Yoder found in their paper, Comparison of Likelihood and Bayesian Methods of Estimating Divergent Times, found that separate analyses often present themselves as unreliable. They rather emphasize a combined use of analyses, as divergence times prove to be the most robust when using the maximum likelihood and Bayesian analyses, multiple genes, and simultaneous use of multiple calibration points. This technique is then used in both Ward and co-authors' 2010 and 2014 papers, where they elucidate the phylogenetic trees of the subfamilies Dolichoderine and myrmicine, respectively. They found that higher partition data sets often confer higher likelihoods than do less partition data, placing more reliability in estimating the times of divergence calculated by the Bayesian analysis. This brings into question the robustness of Wheat and Wahlberg's data, as they may have bit off more than they can chew with such a bold data set. Their data set size may be the underlying reason why their data fails to corroborate findings with other studies surrounding the origins of Arthropoda. In order to verify the findings of Wheat and Wahlberg's study, the use of the likelihood analysis is expected. However, it is understandable why the maximum likelihood analysis was omitted, as computing hours were already nearly 400,000 hours, and its addition would have been extremely unreasonable. However, the verification it provides only further concretes their claim. The use of the Robinson folds metric could help them judge the quality of their phylogenetic inference. In the paper by Wertheim and co-authors, they found that the rate distribution should not be the only factor in determining whether a maximum likelihood or a Bayesian inference should be used, and that a Robinson folds tree-to-tree distance could help determine the quality of their phylogenetic relationships. However, there is a possibility that the use of the maximum likelihood method may weaken Wheat and Wahlberg's findings, as the enormous data set may be unable to be partitioned down into further subsets of data while retaining all 8 calibration points. The only way to implement the technique is to reduce the number of taxa in the dataset and to see if a skeleton outline of the phylogenomic tree mirrors what was originally discovered. This may bring the estimated date of emergence of Arthropoda closer to what Regier and co-authors study found, as the unrestricted age of the root in Wheat and Wahlberg's study may have led to a gross overestimation of the time of divergence. This concludes my presentation. Thank you. Now, in the sort of like self-reflection, I do recognize there is still much to learn in the field of phylogenomics. As for example, I still need to learn more about the different statistical tests and programs are employed in creating tree topologies. The Bayesian method, for example, has many different terminologies that at first glance is difficult to interpret, such as the highest posterior density, but it was easier to understand once it's applied along with the data itself. As for example, it outlines the range of confidence that the data may inhabit. And the aforementioned most likely will come with exposure, as I know that phylogenomics is integral to understanding evolutionary biology in a whole and will most likely come back in my future careers.